Hello, everyone. Thank you for joining us and welcome to the Meet the Wrigley Faculty webinar series. I'm Katie Shavastel, Development Director for the Friends of the Wrigley Institute. Today we'll be virtually exploring the waters surrounding Catalina with two very special members of the Wrigley family. Both of today's presenters trained at the Wrigley Institute, Dr. Dave Ginsberg as a Ph.D. student and Dr. Diane Kim beginning as an undergrad. I'm sure you'll have plenty of questions and we'll answer them at the end of the webinar. So until then, please enter them in the Q&A box throughout the presentations. And please indicate who your question is for. Today's webinar is being recorded, so if you have to leave for any reason or have any technical issues, it will be emailed to you. And without further ado, I'm going to pass it over to another very special guest, the new director of the Wrigley Institute, Dr. Joe Arvai.
Hopefully I'm not, I'm not muted. Thanks, Katie, and hi, everybody. So let me start by adding my welcome to everyone as well, and then also just telling you how excited I am to be the incoming director of the Wrigley Institute. If I could, just for a second, I wanted to thank everybody at the Institute, staff, faculty, for all of their support in helping me to transition to USC. With that, I now have the pleasure of introducing today's speakers, starting first of all with Dr. David Ginsberg. Dr. Ginsburg is an associate professor and he's director of the undergraduate studies in the environmental studies program at USC Dornsife. His teaching and research focus on environmental science, which includes hands-on experiences in the classroom and in the field. He also serves as the USC Assistant Dive Safety Officer here at the Wrigley Marine Science Center, which is a tremendously important role for us. He earned his Ph.D. in marine and environmental biology from the University of Southern California, and he's got a lot of interesting things to tell you. So without further ado, thank you, David, for joining us.
Thanks very much. I'm going to go and share my screen. So we will go ahead and start. All right. So I think we're ready to go. Can everyone see that? Can you guys see that there? Great. Okay. So thanks for having me. It's kind of a really fun project to talk about here. The specific project is looking at marine biodiversity and species richness, or getting a sense of how many species exist in a certain location, in this case doing it where the Marine Lab's located off Santa Catalina Island, just Catalina. This work is basically what I'm talking about today is really part of a larger study that I did with an undergrad who's now actually a Ph.D. student at the University of Florida at Gainesville. Her name was Audrey Luby. She was a NSF REU student at USC, and she was an environmental studies student, scientific diver. And our manuscript was accepted for publication over the summer, so it should come out soon, and that's what I'll be talking about today. So I'm excited to do that. And before I start as well, I have to say a project like this, really any project, but this one in particular, really quite a bit went into this as far as funding, facilities, and then getting information. As far as funding and facilities go, it's really, the Wrigley Institute that was really gracious in, letting us come out there. We did a lot of diving, a lot of time spent at the island. Sea Grant at USC helped provide some money. NSF with National Science Foundation provided some money for student Audrey, and then overall at USC and Dornsife. And as far as species data is concerned, really took a lot of people to get some of the information that I have, and they were very generous in sharing those data with me. So I was really pleased with that, and the Reef Check and the Marine Multi-Agency Rocky Intertidal Network and Vantuna Research Group, and some other folks here. In particular, Andrew Huang worked with me over the summer, who's another ENS undergraduate, and we added some information to this. We did a virtual mentoring-mentee relationship over the summer, and I thought that went really well. So, and I also should probably say, hi, Mom. Hope you are with us today. Okay, so here we are. And to start off with, give you a sense, I think maybe a lot of you aren't necessarily, you might know Catalina, but you don't necessarily know where we fall within the larger picture. And the area that I have shown on the map here is known as the Southern California Bight. Essentially stretches from Santa Barbara to Point Conception, off Santa Barbara to Ensenada, Mexico. And I highlighted those there. So it's a pretty large area, extends out into the water. And there's a lot of different research that goes into looking at the Southern California bight. Where it's important today is what, in relation to what I'm talking to you about, it's a really unique circulation zone. It's the pattern of ocean currents that come through the Southern California bight. You have cold water coming from the north that's moving from the offshore area, and you have warm water coming from the south that's moving near shore. And then these slowly come together to converge in this area. And if you notice, Catalina is essentially in the center of all this. So Catalina is a really unique environment, and my colleague Diane Kim will talk about that too today. As far as kelp goes, makes Catalina a really pretty amazing and unique place as far as the species you find there, and some other qualities about the island. And what's interesting also about the Southern California bight is that it's what's known as a marine biodiversity hotspot. There are a number of these in the world, and right here off Los Angeles, 10 million people living here. We have this really amazing, biodiversity hotspot here. And when you think about how many species you might find in that area, there's a range of numbers, but most people generally, on a somewhat conservative approach, might say at least 6,000 different species of different types of macroalgae, seaweeds, fishes, invertebrates, and that doesn't even include the marine mammals and birds and other types of things that you find there. But this is a really very robust and diverse area, and Catalina plays a big role in that. So, I just told you there are about 6,000 species that exist within the Southern California bight. That might actually go up to 10,000. Not all of those have actually been described, but I am going to try this out here. Our friends at DTS here set up a poll for me, and I'm hoping that we could put a poll up for the number of species that you guys in the audience might think you would find at Catalina, just on the island itself. I told you what's in Southern California bight, and all you have to do is basically click on this poll here, and you can come up with a number, and we should be able to see that. So we can take a minute to do that. I don't think I can vote. We'll give it about a minute. You can just click on your screen, and then as soon as we have an answer, maybe give it maybe 30 seconds or so, we can take a look at what that is. And like I said, this is a really unique area, so it's probably, if you're thinking that Catalina has a lot of species on it, you're right. But the question is, how many, and how does it relate to what's known from the Southern California bight? And I do have a little bit of a method here for why I'm asking this, because a lot of the objectives of this study really kind of relates to that. I'm not sure, do we have an answer yet? Can we maybe just put up what people have put up so far? Okay. I'm not sure, maybe when that, oh, there we go. So thank you. Great poll, thanks so much. So looks like most people have said around 3,200. Some were in that 4,600, a couple people in that 275. I can tell you is definitely more than 275. And if you said 2,400, even probably 3,200, that's close, but 2,400, it's actually about 2,200 species that we find at Catalina, at least of what's been cataloged. So Catalina has about 2,277 species to be exact. That actually includes marine mammals. And that was done by Jack Engel in the late 1970s, did a study on that. It was never published, but amazingly, no one's ever gone back to look at it again. And that was for everything around the island. And that makes Catalina a really unique place for a lot of reasons. One is it has tremendous terrestrial biodiversity. It also has tremendous marine biodiversity, and it's a really unique environment because of that oceanographic transition zone, and a lot of other things. But as we know, Catalina is also, it's part of the Southern Channel Islands. It's one of the largest, or it is the largest of the Southern Channel Islands. It's the only island that in the Channel Island archipelago that has a civilian population of permanent residents of about 4,000 people, including Abalone and 2 harbors. It's about roughly 20 to 30 miles off the coast of LA, depending on where you start. And, you know, people like to go to Catalina. It gets a little bit over 1 million visitors per year. And with that number of people going out to Catalina every year, the environment is under stress. There's pollution, there's people, there's fishing, there's, you know, a number of ecosystem stressors on the island. So I think, you know, in being able to say, hey, we have a number of species out there that's, you know, significant. It's, you know, roughly half or so of what you might expect for a Southern California bite. I think it lends itself to ask the question of, well, how many species do we have in our protected areas, and how many, you know, do we know about, and are there more that we've never seen? So that's kind of one of the reasons for looking at this study. In particular, I'm talking about these protected areas, so an MPA, and I'm talking about Blue Cavern, Onshore State Marine Conservation Area. I have it highlighted there. It's almost 7 square kilometers, and it's among one of the oldest, and it is actually one of the larger MPAs that we have in Southern California. It was first established under a different name in 1988, and then it was kind of revamped in 2011. But it's been around for some time. It's one of 9 marine protected areas that you can see here around Catalina, and there are a number of them. But what's unique about this one is that it's one of the largest and one of the no-take zones, meaning you can't take anything out of the Blue Cavern SMCA, and you can't anchor there, and you can't fish there, and you basically have to have a reason to be, you know, doing activities inside of there. Within some, there's a little bit of reason there, but yeah, it's a really kind of unique place, and it's a very very beautiful place. And you know what's amazing about this, given that this has been a protected area since 1988, labeled by California, is that there's no comprehensive species list for this area. So, you know, that brings up the question of, well, how many organisms are out there? And from this study, I can tell you, you know, the punchline is that there's about 1,100 species that within the Blue Cavern SMCA, you know, and I think this is a great baseline for understanding species richness, and I think it will help to improve management and conservation of other areas in the Southern California Bight. So from here, I wanted to kind of give a little bit of an overview of some of the highlights of the species that we found. It's a tough job, had to do a lot of diving, about 70, 80 dives, at least 60, 70 hours spent underwater. Audrey and I pretty much dove every day for a couple summers, and we got a lot done. We looked at the literature. We did a number of things. I won't go into the detail of the methods. If you have questions later, happy to answer that. So I have a few categories. So here's one. In the upper right corner, it says N equals 18. That's the total number of what I would call invaders or exotics. Couple are highlighted here. Essentially these, particularly Sargassum or water cypra or the Diplosoma, these compete with native species. They encrust things, they foul boats, they foul docks. They are not really a good thing, although there's a lot of questions about how bad are they and will they become worse. But these are invaders, these are invasives, they're not from California, and there are currently a total of 18 of these in Blue Cavern. These are the kind of more remarkable ones, but the brown alga Sargassum has posed some issues with fish and competing with giant kelp, Macrocystis. So that's a lot of studies going on with that. A couple others I added in here. These are kind of interesting, some climate busters, and a big collaborative project with colleagues at USC, Sergey Knudsen and others. We were looking at the potential for methane reduction of Asparagopsis, which is a red alga, and Zonaria, which is a brown alga. And essentially what you can do is take these, grind them up, add it to cattle feed, and cows will eat this. And it turns out that they don't mind the taste, and it actually reduces the amount that they burp and the amount of flatulence they have, which releases quite a bit of methane. And this could be something for the future. And we have these right here in Big Fisherman's Cove and all around the reserve. Number of species here that I would consider as part of the the visitors and survivors. Again, there are about 14 of these, what are sometimes known as range extensions. These guys should not be here, at least the ones at the top. The one at the top left is a largemouth blenny. The one on the top right is a fine scale triggerfish. These are both, I've seen the triggerfish right underneath the dock at USC. And these guys are visitors. They essentially started in Mexico, warm water events like in El Nino. They can take advantage of that. And then lo and behold, they end up at Catalina and they never leave. There's some questions about whether they're viable, meaning can they actually reproduce and things like that, because they might not have others around. But they certainly have been around for a long time, and there's actually some butterfly fish that used to hang out in off the cove. And I think there actually was a great postcard at one point that I found at the general store saying, "Welcome to Catalina," and it showed 2 butterfly fish, which definitely do not belong off the coast of California. They were from Mexico. And then the one at the bottom is a blue dorid. That's a nudibranch, really beautiful organism, about as long as about half the length of your finger. And those BFC is Big Fisherman's Cove. It's one of the last vestiges of their range. They used to be found throughout Southern California. It's thought that maybe through water pollution and some other anthropogenic impacts, they have been on the decline. And one of 2 places that you can find them is actually in the Big Fisherman's Cove in the Blue Cavern Onshore SMCA and State Marine Reserve. So yeah, it's really kind of a rare thing. And when you see these, you know it. They stand right out, and beautiful little creature. Colonizers, and I don't mean by the person there, which is actually me, but seagrass. And so eelgrass is believed to have come from the Northern Channel Islands, probably from Anacapa, sometime in the 80s probably. It was first documented off Catalina in the 90s. And with the seagrass, which is really interesting, is if you look at the accounts prior to the 1996 when it was first documented, you didn't see some of these organisms. And there are more than I have here, but these large stingrays, the big bat rays people like to look at, they really like the seagrass. They eat things there. Round rays, little orange-throated pike blenny. These are things that kind of came along with the seagrass. They didn't necessarily colonize, but they're organisms you find there. So seagrass is really important. It's an important ecosystem service and really important for nurseries and essential fish habitat. Some guys have been missing in action. These 2, Batstars, spiny stars, they used to be quite common. Now they're pretty rare. Mass mortality is probably through El Ninos and a viral pathogen. So sea star wasting disease, really don't see these very much in the cove. And looking back through some of these data, it's interesting to see in the 80s they were there. And after that, we just don't see them anymore. I've actually never seen a Batstar in 20 years of diving in the area around USC. I have seen it on Catalina once. And then finally, some of the vulnerable and endangered organisms. Abalone, which generally, those of us in our, 40s or more know abalone. Kids today don't really know abalone. It's a large herbivorous gastropod snail. Black abalone, of course, are endangered, as are white. Green and pink abalone are at risk and vulnerable, although the green abalone seem to be coming back. You can see a pile of abalone shells off Santa Barbara in the 1900s. You'll never see that again. Yeah, abalone have had a tough time, and some of that is due to wasting disease, which has affected them, and some of that is due to overfishing. And they really kind of took a double whammy back in the 90s and early 2000s. And then, of course, the return of the king. This is the giant sea bass. And what's interesting about these guys is they were common, in the throughout the 40s and before, in the 50s, and starting in the 60s and 70s, they were really on a decline, and the fishery was declared essentially collapsed when you couldn't find them anymore. They do exist in Mexico. This is a pregnant female. It's probably about 5.5 feet and probably in the 600-pound range. And that was in about 20 feet of water off the dock at USC at the Marine Lab. It's the largest bony fish in the kelp forest. It's considered endangered, although on an international level, it's considered critically endangered. And these guys are actually making a comeback. And these had never been documented in even though people have seen them, they've never actually been officially documented off the protected area, off the Marine Lab. And that's something that I was kind of proud that we were able to say, hey, they're here, and we see them quite a bit. And just kind of finishing up here as far as a summary and some conclusions is that, as I said, there are about 1,100 different species of seaweeds, fishes, invertebrates that have been documented in Blue Cavern onshore SMCA. And that's a good amount. That's about half of what that's about 50% of what we know from Catalina. And the total species that were recorded, what's interesting is it's about 60% of the estimated richness. And I didn't go into those stats, but if you estimate how much you might find, it's about 60% of that, which suggests that there could be as much as 40% of the species that are missing or yet to be reported. And I think that's a really interesting number because, you know, is this something that, are we truly missing 40%? Have we not looked in the right place? Are we counting the wrong things? Or are they just not there? And I think I'm pretty confident of that 40% number, and I can talk about that in the questions later. Overall, you know, I think investigations like this, they're not just stamp collecting. I think it's really important to know what you have and to get these estimates of species richness. And it provides a lot of important insights for resource managers and you know, biologists that are interested in, you know, connecting the dots between spatial and temporal patterns of organisms, not just at Catalina, but the other Channel Islands and Southern California Bight in general. So with that, I'll finish, and thanks for listening. And I'll take some questions at the very end. Thank you.
So thanks a lot for that, David. I'm not in control of my video, I don't think. I've just been asked to start my video. This is so much fun, Zoom. Hopefully you can all see me now and hear me. So thank you again, David, for that. And, you know, as I was listening to David talk, I was also looking at the attendees. We have a very large number of people on the call with us today. So if you're out there in the audience, thank you so very much for coming. And if you're a student, welcome back to USC and hopefully to campus real soon. So I have the pleasure now of introducing our next speaker, Dr. Diane Kim. Diane is one of our research scientists, and she supports multiple research projects within the Wrigley Institute. She also designs and leads research, training and education programs for our undergrads at USC. And in addition to all that, she's an adjunct professor in environmental studies at USC Dornsife. And then one last thing, if that wasn't enough, Diane is also the Wrigley Institute's inaugural coordinator for diversity, equity, and inclusion, which is one of our new priority areas at Wrigley. So a huge thank you to Diane for taking on that important role. She earned her Bachelor of Science in Biological Sciences with a minor in Business Administration, and then her PhD in Marine Environmental Biology at the University of Southern California. Without further ado, welcome, Diane.
Thank you so much, Joe. I'm going to share my screen now. Okay, hi everyone. Again, thanks. I just want to echo Joe's thanks to you all for joining us this afternoon. I'm Diane, and like you said, I'm staff at the Wrigley Institute and adjunct faculty in environmental studies. I'm also co-founder of Holdfast, let me turn my camera, of Holdfast Aquaculture, where we provide resources for ocean farmers. And the title of my talk today starts with Under Pressure, because I'm going to tell you about a novel strategy that we're testing to cultivate kelp. This is a strategy that involves physically moving kelp between near the surface of the ocean to much deeper depths that expose them to much greater pressures than they're normally used to. And I'll describe why we're using this strategy and why we're looking at kelp to begin with later in my talk. But first, I want to mention that this is a project that's funded by the Department of Energy's arm, ARPA-E, Advanced Research Projects Agency on Energy. And I have to say that it's a pretty fun and exciting project to work on because of its applied nature. Right? We're working on, you know, the very early stages of something that could one day disrupt the energy sector as we know it. By providing a cost-effective source of renewable bioenergy to displace fossil fuels. I think this project is also a really great example of what can be achieved through industry and academic collaboration. So Marine BioEnergy is our industry partner. The co-founder, Brian Wilcox, is the principal investigator and engineer on the project. The Wrigley Institute, we bring the biological and oceanographic expertise, and we also offer a platform for testing these types of innovative solutions. And the Pacific Northwest National Laboratory, they're the chemical engineers focused on the downstream end of the pipeline where you're converting the kelp biomass into a usable form of energy. So full disclosure here, I'm an oceanographer and microbial ecologist by training, but since 2017, I've had the privilege of getting to really dive into the world of kelp, both figuratively and literally, as you can see here in the photo of me on the right. This is my dive buddy and colleague. He's a postdoc at the Wrigley Institute, Andy Navarrete. He's been instrumental in the success of this project thus far. And behind us, you can see the target organism. This is Macrocystis pyrifera, or giant kelp. Some people refer to it as the sequoias of the sea because, as you can see from the picture, they form these really beautiful, lush underwater forests. They provide a really important habitat, refuge, food source for a lot of the biodiversity that Dave mentioned in his talk. So they're really foundational species for marine ecosystems. They also provide really essential ecosystem services. They sequester carbon dioxide, and so they can buffer against the impacts of climate change, including ocean acidification. They literally breathe life back into the oceans because they produce oxygen as a byproduct of their growth. And this particular type of seaweed or kelp is typically found in these shallow subtidal environments. It's attached to rocky substrate like you see in the photo here. And they can get pretty big. An individual can get to be over 100 feet long, which is pretty massive. And each one can be comprised of many of these fronds. So here's a picture of one frond, which is essentially a stipe or a stem with all of its associated blades or leaf-like structures here. And you'll notice on this frond, at the base of each blade, there are these bulbous structures. These are called Pneumaticists. They're gas-filled and they provide buoyancy for the blade to present themselves better to light. So despite the ability to get really large, they actually start off no bigger than a large bacterium. So they start off as these little tiny spores that are swimming around that develop into either a female or a male. They produce sperm and egg, producing little tiny baby microscopic kelp. And they all start off as a single-bladed individual before growing into these massive ecosystem engineers, if you will. So just to, I told you that the title of my talk, Under Pressure, that it alludes to this depth cycling strategy that we're testing. But it also alludes to the mounting societal and environmental pressures of continuing to rely on fossil fuel-based sources of energy. So here's a pie diagram of energy consumption by energy source in the United States in 2019 by the Energy Information Administration. And what you can see is that renewable energy makes up a very small fraction of this pie, 11%, while fossil fuel-based sources make up 80%, bigger chunk, petroleum, fossil gas, coal. And these are things that continue to exacerbate global climate change. And so the driving force behind this project is to try and change the size of these pie slices from a fossil fuel dominant energy infrastructure to a more sustainable alternative. And we think kelp might help shift that energy landscape by providing a sustainable feedstock for biofuel production. We're talking about a relatively carbon neutral source, especially to fossil fuels that took tens to hundreds of millions of years to form, only to be extracted in a couple of centuries. Instead, we're talking about something, that yes, when burned, it will emit carbon into the atmosphere, right? Kelp oil will emit carbon into the atmosphere, but you also absorb a lot of carbon as you're growing the kelp to begin with. So it's a more regenerative model rather than an extractive one. We also understand the life cycle really well, so we can grow it in a nursery setting for outplanting for farms or experimental purposes. And we understand it so well that we can preserve them in their microscopic stages of life, and have, we can also have an understanding of some of the traits that will result from growing those individual cultivars. And that's another project that we're working on with Dr. Sergey Nuzhdin at USC that I can talk about next time. Kelp is also one of the fastest growing organisms on the planet, right? We're talking about documented rates of over a foot per day. So you can literally watch kelp grow. They're easier to digest than land plants because unlike land plants, they don't have to fight gravity. So they don't need these complex compounds like lignin to hold them up. And here's the kicker. You don't need land or fresh water to grow kelp. You grow it in the ocean. And of course, these are resources that are becoming increasingly scarce. And you don't need fertilizer to grow kelp either. There's plenty of nutrients in the ocean that I'll talk about in just a second. When thinking about kelp for energy, you also have to consider, the scale of operation or growing operation that you need to supply all of the, energy demands. Fortunately, there's a lot of space in the open ocean that could be utilized for this. And in fact, the United States has the largest exclusive economic zone, which is the ocean space that spans out 200 miles from the coastline. We have the largest EEZ in the world that could be utilized. But of course, farming in the ocean is not easy. First of all, the ocean on average is about 4,000 meters deep. There's no substrate for kelp to attach to near the surface. And when you're looking at the distribution of the two things that kelp need for growth, light and nutrients, these are things that are typically not found in the same part of the water column. So for example, light intensity is highest near the surface and weakens as you get deeper. And in fact, most of the ocean is a dark environment. Nutrients give you a different profile. They're very low to zero near the surface, but as you get deeper, the nutrient concentration builds. And so the challenge is, delivering both nutrients and light to kelp all year round for uninterrupted production. And there are some groups that are funded by the same agency, ARPA-E, who are testing strategies that involve irrigation of deep nutrient-rich water up to the surface where the kelp are, right? So artificially pumping deep water up to the surface. This is a strategy that people actually looked into back in the 70s as well. But our collaborator had something else in mind. He decided that it would be more practical, I guess, from an engineering standpoint, to take kelp down to where the nutrients are instead of taking the nutrients to where the kelp are. So what he's envisioning are these autonomous, unanchored drones that pull kelp arrays, these kelp farms that are continuously moving throughout the world's ocean, slowly but constantly moving, including taking the kelp down to where the nutrients are to bathe them in nutrient-rich water, and then taking them back up to the surface where they can bathe in light to photosynthesize. So the problem with this is that no one has ever tested the biological response of kelp to depth cycling like this. So last year, so this is where the Wrigley Institute comes in. We helped set up a proof of concept to test the biological response of kelp to depth cycling. Brian Wilcox developed an anchored experimental platform that moved a structure between 80 meters at night and 9 meters where you get plenty of light during the day. We nicknamed this contraption the Buoy Elevator because of that up and down motion. And we planted kelp on this thing to run an experiment. And along with this group, we planted kelp near a native kelp bed at a place called Parsons Landing. Here's a picture of the elevator platform. All of this took place out on Catalina Island that Dave mentioned in his talk. If you zoom into the isthmus here, you'll see Big Fisherman's Cove where the Wrigley Marine Science Center is located. This is where the elevator was deployed in deep water here, and this is Parsons Landing, the site of that native kelp bed. So those are the two experimental sites. And all the kelp started off really small, really young, all single-bladed, right? So very young. And on average, they were about 25 centimeters or 10 inches in length, and we outplanted 35 individuals at each of the sites. So we let the experiment run for about 100 days, and here are a couple pictures. On the left here, this is the elevator site, the deep water site, where you can see very clear blue waters at the surface. This is where the kelp were cycled daily. And then on the right here is Parsons Landing. You can see the kelp in the backdrop here and the line where we outplanted the small juveniles. We had a temperature logger on this elevator structure, and here's the temperature data from that logger showing you that the temperature fluctuated between, warmer temperatures when it was at the surface during the day, down to colder temperatures near 10 degrees Celsius when we cycled it down to 80 meters. And you can see the up and down, daily cycling for the duration of the experiment with a few interruptions because of large swells and technical glitches. So some of our leading questions included, will kelp grow, let alone survive under depth cycling conditions? We're talking about kelp, taking kelp down to 80 meters every day. And, with every 10 meters, you're adding 1 atmosphere of pressure. And so down at 80 meters, you're talking about quite a bit of pressure change. So we just weren't sure how the kelp would respond. We also weren't sure what would happen to these nematocysts that I mentioned early on. These are gas-filled. And so we were concerned what would happen about what would happen to them. We did some preliminary trials using a hyperbaric chamber on Catalina and found that beyond about 40 meters, the pressure that you experience beyond about 40 meters, all of the fully formed nematocysts would pop, would implode and produce this slit that you see in the picture, and then the tissue would get really soft and we would lose the blades. So that was a problem and a concern. It was also really important for us to collect some evidence to show whether kelp could take up nutrients in the deep, dark, cold environment separated from light at the surface, because there's some evidence that certain seaweeds can't do this or have limited capabilities of doing this. So to cut to the chase and answering the first question, will kelp grow, let alone survive? This is the elevator site, a video captured by Juan Aguilar during a free dive. He is also a Wrigley Institute captain and has been a vital part of this project. And remember, these kelp started off as single-bladed individuals, about 10 inches in length, and you can see that they not only survived, but they grew quite a bit. And you can also see the consistency between the replicates at this site, which is really cool. I hope that didn't make anyone seasick. The point is that the depth cycled kelp survived and actually grew faster than kelp planted near a natural kelp bed. So at the end, the kelp that were depth cycled were actually on average 4 times heavier, depicted by this bar graph here. They were 4 times heavier than the individuals that were recovered from near the native kelp bed at Parsons Landing. Here's a picture of one of the largest individuals that were depth cycled. Remember, it started off a little smaller than the size of this yellow ruler. At the end, it was about 6.3 meters long. So what will happen to nematocysts? Turns out that they will actually develop nematocysts as they grow, but they look different from what you might expect. So they're smaller, they're not as wide. These are all the nematocysts from the individuals that were depth cycled. So they weren't as wide as the ones from the native kelp bed. They were also much more variable and a bit longer in blade stem length, which you can see in this picture, which is basically the part that connects them to their main stipe. So these were much longer than what you might expect from a natural kelp. When we sliced these open, these nematocysts open, we also saw that the wall thickness was greater for the depth cycled individuals compared to the ones from the native kelp bed. And a lot of these were also fluid filled, interestingly. So will kelp be able to assimilate nitrate from deeper waters? We addressed this question by analyzing the tissue composition of kelp. And what we saw was that the depth cycled individuals had a greater protein content shown in this purple slice here than those individuals recovered from the native kelp bed near Parsons Landing. And you can't have protein without nitrogen. When we looked at the total nitrogen content, this also confirmed the fact that these depth cycled kelp were not nitrogen deficient. They in fact had more nitrogen than the individuals from near the native kelp bed. And we used nitrogen isotope data, which can basically tell you a little bit about where the nitrogen comes from, the source of that nitrogen. And for the purposes of this talk, I'll just say that this information told us that not only are the depth cycled individuals, not only did the depth cycled individuals show us a distinct or unique nitrogen isotopic signature, but that signature suggests that they obtained their nitrogen from a deeper source. So, based on these summary results, I think, I'm happy to say that depth cycling seems to be a viable strategy for growing kelp. I think there's still a lot of work to be done and many questions to be answered. But this is a significant milestone because it begins to address one of the biggest and most fundamental challenges in the kelp biofuel pipeline, which has to do with figuring out how to consistently deliver nutrients and light to kelp for year-round, so uninterrupted production and large-scale production of kelp. So if we can figure out the technology and logistics of how to do this cost effectively, then we might be onto something here. So I want to end by thanking all the contributors, so many contributors to this project, which is ongoing, including sponsors, collaborators, especially Dan Reed, regulatory agencies, everyone at the Wrigley Institute, all the co-authors. We have a paper coming out, so you can consider this a preview of that paper. Dave Ginsberg is a co-PI on the project, so you can direct all the challenging questions to him at the end. I'm just kidding. And we're so fortunate to work with a large number of very talented and motivated undergraduates who came to us from various sources, funded through various programs like the National Science Foundation's REU and IU's research training programs, the Environmental Studies program, and a special shout out to the Zinsmeier family for their continued enthusiasm for kelp biofuels and undergraduate training programs. So with that, I'm happy to take questions. And hopefully I was not muted for the whole thing, like Joe always says.
All right. Well, hi, everybody. I'm Ann Close, the Wrigley Institute. And Dave, Diane, thank you so much for that. Excellent. Excellent. Dave, start off with a couple of questions for you. Can you talk to us a little bit about the scale of the things you're looking at? Is everything visible? And what, sort of a second part to that, what's known about the microbial diversity in the area?
Yeah, so the first part is the size was about 1 centimeter or larger. So that was kind of our metric of what we could see. So it was really visible things, which is, there's probably quite a few things that are smaller than that you can't easily find by just doing scuba surveys. But looking in the literature, there are people that have gone in and looked at very specific size classes smaller than that. And so we were able to capture some of that. But yeah, as far as visual surveys go, it was 1 centimeter or greater. And as far as microbial diversity, we didn't look, we basically cut things off we didn't look at any microbes. So we didn't look at bacteria, viruses, anything like that. And we actually kept protists out of the study too, because I think that would add quite a bit. And it was more than we could handle at that point. But there is a lot. I'm sorry, what's that, Diane?
Kelp are protists.
Yeah, no, I love protists. But there's a lot known about microbial communities. I can't answer it right now, but there's quite a bit known about them especially off Catalina and in the channel.
Great, thanks. So it was exciting to hear about how much more there is to be discovered and how much more we have to learn. Can you talk a little bit about some of the documentation methods that are used, and are some things you might be looking forward to in the future?
Yeah, I mean, I think the way we did it the slow and painful way, which is to go out and search through the literature and do scuba surveys. There's a group at UCLA with Paul Barber's group at UCLA who's been using eDNA, and they have a really pretty neat method. It's a Anacapa protocol, and they started it off Anacapa. A couple students and now postdocs, Emily Curd and Zach Gold, have worked on this. And I think eDNA would be just fantastic to use. But I think you also need to go out and, you need to take that painful way to know what you need some kind of comparison, some kind of baseline. So I think there's a little bit of both. Now that we've counted things, it would be interesting to do the eDNA part using molecular methods. But I don't know of any other studies that have done this off the Channel Islands. So yeah, I took the painful way, but I think, would I do it again? Probably not. And when I asked Jack Engel, who did the first study in 1978 about this, he said, yeah, it almost killed him and he would never do it again, which is why I think, no one has done it in so long. But yeah, I think that's my answer there.
Great. Can you tell us how, it's cool to hear about the invasive species. How do they get established?
Well, they can be established a number of ways. I mean, usually it's through some kind of, might be through ship ballast, it might be through some kind of commerce or other ways that they can become, on fouling on someone's, boat or other, something else that's in the water. And they can basically, take root and just start to go. I mean, some things actually can move with currents, and the range extensions that I talked about with the triggerfish and some of the others, I mean, in a way it's an invasion, but it's officially known as a range extension because while they're not from here, they're not that far away, but some things can actually travel pretty far. And I think as far as the bryozoan and the Ascidians go that I talked about, I'm not sure anyone knows specifically how they got here, but I could be wrong. But I think that is a big question sometimes. How do they get here? And there are often cases where we don't know.
So also really interesting to hear about the brown and red algae and the cows.
Oh yeah.
Cool. Can you tell us about the mechanism? Like how does that actually reduce the methane? Or do we need a whole semester of chemistry for that?
Yeah, thank you, Will Barrelson, for that question. But I mean, it's essentially, there are some organobromine compounds that have been identified in marine algae, and those include things like bromomethane and bromoform, which you can actually buy bromoform, and it's used in some industrial processes. But those are identified as being able to inhibit microbial methanogenesis. And so, and remember, this hasn't been on a commercial scale yet, so it's all a lot of in vitro testing. So some of it's hypothetical, but it seems as though those would actually inhibit the methane production. But, in talking to a chemical ecologist about this, they said, you're crazy. I mean, those bromoforms we have to be careful about that if you're eating, just trying to pickle seaweed from the water. I mean, those bromoforms could kill you. So I'm not sure how the cows get by. Apparently it doesn't affect the taste of the milk from the dairy cows, but it's kind of surprising. And the chemical ecologist I talked to just said, there's no way, never, can't happen. And, other chemists at UC Davis said, oh yeah, it happens all the time. So yeah, it's a little bit beyond what I do, but that is essentially the mechanism, those organobromine compounds.
All right, I'm going to throw this one out to both of you. We've had a couple of folks ask, as undergrads, how can they get involved with research? Dave, you talked about this. Diane, huge part of the Kelp project. So if you guys can both chat about that a little.
I think that's Diane, the good spot for Diane to kick in there.
Yeah, thank you for asking that question. I mean, we're always looking to engage undergraduates who are really motivated by these types of projects. So, during the semester, USC students can work with us through a few different channels. You can work with us through courses, so you can get units, 2 or 4 units, and participate in various research projects with us through things like the 490 course, directed research, or 492 through the environmental studies program, the environmental internship class. You can also look for funding opportunities. So things like SURF and SOAR, these are fellowships that are offered through the Office of Undergraduate Studies at USC that give you a stipend to work on a research project with a faculty mentor at USC. We also have, volunteer and internship opportunities throughout the year as well, which is a great way to get your foot in the door. During the summers, that's probably when we're most active with research activities, especially out on the island. There are various channels then as well. So we have a couple of, one program that's funded by the National Science Foundation, the REU, or Research Experiences for Undergraduates. We're always looking for students to work with us through that program. Applications typically open in January of each year, so you can keep an eye out for that. This is a program that basically pairs you with faculty and near-peer graduate student mentors to work on independent research out on the island. Usually, this year we ran it virtually, which was interesting, but I think it went well. There are also internships that are offered during the summer. The Zinsmeyer family supports up to 4 to 6 undergraduates every summer as well. So, lots of different ways to engage with us. And I think the best thing to do is just come and talk to us, shoot us an email.
Great.
Yeah, I'd agree.
Sorry, Dave, did you want to add something to that?
No, I was just going to agree with Diane. I think that's exactly, contact us and come talk to us.
All right. Well, some more kelp specific questions. With these open ocean farms starting up, what are some of the effects, positive or negative, to the surrounding areas and to the species around there? Do we know much about that yet, or?
Yeah, that's a really good question and a really important one that we have to think more about. It's something that is, on our radar, but we haven't thought too deeply about yet because we're just at the early stages of all of this, right? But if you're talking about cultivating kelp on a massive scale in places that kelp normally is not found, right? In the open ocean, for example, knowing that kelp provides such an essential habitat, food source, et cetera, for so many different marine species, it's not unreasonable to think that you're going to attract a lot of other things out to these kelp farms in areas where they don't belong either. And so these, unintended ecological consequences are real. It's something that we have to think about, but we just haven't gotten there yet to do a deep analysis. But it's something that that we should think about more.
And sort of following on that and talking about consequences, when you burn kelp, aside from getting CO2, do you get things that we need to be concerned about? Sulfur oxides, anything else that should be keeping us up at night?
Probably, I don't know. Dave, do you know?
No, I don't know. I mean, they used to burn kelp, potash and things like that, but I don't know. I saw that question and I don't know.
Oh.
All right. Oh, go ahead.
What I will say about Kelp Energy is that as far as I know, the method that our collaborators are looking into is something called hydrothermal liquefaction that would take wet biomass and convert it into a crude oil and some other products. But that crude oil is something that can be fed into existing refineries. And in fact, I read a couple of articles recently talking about the surplus of refineries and how some are actually making that switch to become a refinery for bioenergy purposes. But I thought I'd throw that out there in case anyone was interested.
Dan, can you talk a little bit about the how the Kelp Elevator is powered?
Yeah, so the elevator is powered using solar panels that are attached to the sides of 4 walls that are mounted on a buoy, and inside the 4 walls is a winch that's automatically programmed to raise and lower the submerged structure with attached kelp, and that's all powered using the solar panels. Now, when the open ocean autonomous farms that our collaborator Brian Wilcox is envisioning has nothing to do with this elevator structure, right? Because he's talking about unanchored things that are constantly moving throughout the ocean. This elevator platform was just a proof of concept just to test the biology, right? How the biology was would respond to this up and down motion every day. So for the open ocean farms, he's envisioning the use of wave energy combined with solar energy for powering these underwater structures. And you'll have to talk to him directly for more information on that.
Do we know yet if this is economically viable?
I mean, that's a really important question, because we have to ultimately beat, whatever, $3.50 a gallon at the pump, right? Not only do we have to produce enough to meet all of our energy demands so that we can continue driving our cars and charging our iPhones, but it has to be cheap enough. And, part of that will depend on, the materials and the scale of operation. And these assessments are, being done by our collaborator at Marine BioEnergy. They're doing a life cycle assessment on this technology. So I'll have to defer to them for a more specific answer than that.
All right. Have you thought about moving the Kelp Elevator at different rates? Like how was that decided on? What effects does that have? Do we know those answers?
That's such a great question too. So initially for this first experiment, we wanted to maximize the amount of time at depth where they're exposed to a lot of nutrients, and maximize the amount of time up at the surface where they have plenty of sunlight. But, in thinking about saturation of both nutrients and light, you might start to think about how to optimize these depth cycling parameters so that maybe instead of cycling just once up and down per day, you're cycling a few times a day. So there are a lot of these things that we're wondering about as well, not just the rates, but the actual depth that we go down to, how often do we go down? These are all really important questions that could ultimately lead to increased biomass yields. So it's something that we're looking into, and we are running another experiment. This darn COVID thing is, slowing things down, but we do have plans to run another cycling experiment where we'll be addressing some of these questions.
Great. Can kelp be used as a carbon source for methane digestion?
Kelp for methane digestion?
For digesters.
Oh, for digesters, yes, kelp, yeah. So I only mentioned hydrothermal liquefaction for the production of crude oil, but certainly you can use things like anaerobic digestion to produce methane. Is that the question? Which can be used to generate this natural gas to feed into that natural gas pipeline. You could also ferment the biomass to produce ethanol. Right now, the gas that we pump into our cars is being supplemented by ethanol, about 10% ethanol, all of which is derived from corn, right? So, I mean, there are a few different pathways for kelp to be used as an energy source. Was that the question?
That was the question.
Okay.
All right, and I just want to be respectful of our time. And so I see it's 3 o'clock. Thank you both very much. And with that, I will pass it back to you, Katie.
Okay, if DTS could put my video back on, start my video. Awesome, okay. Well, thank you so much, everyone, for all those amazing questions. And thanks to Dave and Diane for guiding us through Catalina's unique marine habitat and sharing its potential. If you have any further questions or we didn't get to your questions, please feel free to reach out to me. I'll be sending out an email with my contact information and a link to the webinar recording. If you'd like to check out any of our previous webinars, the series is hosted on our Meet the Wrigley Faculty YouTube playlist. You can find the URL in the chat box. Our next webinar will be on September 16th with Dr. Shannon Gibson. She'll be discussing the Paris Climate Agreement, a climate justice take on the treaty, and COVID-19's impact on global climate negotiations. Keep an eye on your inbox and social media for the invitation. So again, just thanks for joining us this afternoon, and we look forward to seeing you next time. Thanks, everyone.